I am presenting my senior research project to you guys that I've been doing for the last year. I did get a great opportunity to go to the Cayambe National Reserve in Peru this last summer with a nonprofit organization known as Earthwatch. So was doing some field research down there and saw a great opportunity to couple my senior research project with this. So I developed this project here in which I looked at the effect of deforestation on microbial biodiversity and looking for potential novel species of actinomycetes. So, okay. So we're in the Amazon. Again, I was traveling there to do some field research with Earthwatch, but it is also one of the largest reservoirs for biodiversity on Earth. So not only just plants and animals, but microbial species as well. There have been large areas that have been deforested in the last 40 years in this region. So again, it was also an area of interest to look at the deforestation effects. A few microbial studies have also been done there, which was surprising to me. And so there's many microbes that remain unidentified at this point, and so specifically, again, was looking for actinomycetes. So my first aim to my project was to look at the effects of deforestation, as I had mentioned. I wanted to see if we were killing certain species at the microbial level. Always hear about deforestation effects on animals and plants, but not necessarily about species that you can't see. So I wanted to kind of look at that area and see if we're killing any critically important species at that level. And then my second aim was to isolate any potential novel actinomycetes that could potentially be medicinally important or useful in the field of medicine. So at first glance, after plating, I did do nutrient agar plates. As you can see here, I do have these red squares. VG stands for Village Garden, so I did use slash and burn village gardens for my sample sites for the deforested areas. The Cayambe National Reserve has banned deforestation, so I used the village garden samples to represent the deforested areas. And then if you look over here, you have the FE, which just stands for forest edge, which is my pristine samples. And again, at first glance here, I do see some antifungal ability. You can see, in the circles here, you can see that there is a killing zone there that is prohibiting growth of the fungus around that bacteria colony, which is I had selected to study as fungal infections are important in medicine, in humans and in amphibians as well, such as chytrid fungus or the white nose syndrome that is killing bats around the world. So I did select a couple of the antifungal colonies for study during my project here. I also noticed one single colony on one plate in my pristine sample that had this interesting orange water-soluble pigment being secreted from that colony. Again, I only saw this on one plate, one single colony, so it's very interesting. So I did proceed with this as well in my biodiversity study. And again, I saw different pigmentation within some of the pristine samples that I didn't see in village garden samples. So I did select some colonies based off of color. The yellow circle on the left-hand side shows an orange pigmented bacteria. The blue circle is circling a yellow pigmented colony. And then on the right-hand side there, that is a purple pigmented colony that I had selected for study. As you can see, the black circles, there was multiple colonies of orange pigmented bacteria on all of my pristine plates. This was not present in any of my village garden samples, which I thought was very interesting to see that the orange pigmentation was seen throughout all pristine and not in the village garden samples. So a summary there on the left-hand side shows the village garden samples versus the pristine forest samples and what colonies I selected for what characteristics. So the village garden I simply selected based off of antifungal ability, as I didn't see any other special characteristics that would lead me to select them. The pristine forest samples, however, I did see a lot of pigmentation, so I chose a purple colony, a couple orange colonies, a red colony, and that white secreting orange pigmented colony. Throughout the biodiversity assays, I did do many traditional species identification studies, such as morphology, looking at gram, stain, and size. I did do one behavioral test for mobility, and I did some metabolic tests, including citrase, catalase, oxidase, some of those traditional tests as well. And then I did go on to do some more modern testing with genetic species identification, in which I ran Flemish train reactions, did some gel extractions, and then packaged for sequencing to be sent to the University of Iowa. As you can see on the right-hand corner there, this is one of the examples that I received back from the University of Iowa for my trace sequencing. Each of the different colored peaks represents a different nucleotide, and so I was able to do a blast search based off of what I received from them. So here's a table of my morphological and metabolic results during my biodiversity testing. A couple things to point out. For the village garden samples, I did choose, again, all of the antifungal species here. I did have a suspicion that they were all the same species. They appeared the same, they were the same pigmentation, and they all had that antifungal ability. So kind of led me to think that they were maybe the same species, and as I was going through some of my tests, they do match up fairly similarly across the board, except for one little difference here in the catalase test. It did show up negative, and I did redo it multiple times because I was a little confused as to why it didn't match the rest of the studies, which I will talk about a little bit more in the next slide. And then another thing to point out here, down at the bottom, the purple, you can see here I have an NA. Unfortunately, my purple species of bacteria was not viable in the storage conditions I chose. So about halfway through my studies, I did end up losing this particular bacteria, which again, I'll talk about it a little bit later, so I was a little disappointed about that, but that's why you see the NA here. And then here's my sequencing results that I had received from the University of Iowa. Based off of the nucleotide sequences I had obtained, I did do a blast search and looked at similarity among other sequences that have been identified. The white antifungal ability, that one I pointed out to you guys, that one test did show me that it was something different. For the most part, the other four species were fairly similar to Bacillus megatherium, which is found in many different things, such as foods. It's found in animals and humans. It's used in synthetic penicillin, so used for many different things, particularly in industry. But again, like I said, it was interesting to see that that one test really did show it was a different species. One thing to point out here too, my orange antifungal that I found among that orange pigmented species in the pristine samples, unfortunately, it matched fairly similarly to Serratia martescens, which is pretty similar to my red bacteria. Throughout all of my studies, my red bacteria was contaminating my orange. So I do believe there's a little error here. I think that it had once again snuck in before I packaged for sequencing to the University of Iowa. So I don't believe that it's Serratia martescens, but that's what my blast sequences did match similarly to. So, and then going back to that purple bacteria that I had lost in my storage conditions, I did use Bergey's manual to match up the studies that I had so far, and I came very similar to Genthinobacterium bovidum. This particular bacteria is an aerobic and gram-negative bacteria. It has that purple pigment that results from glycerol metabolism. It is antiviral, antibacterial, and antifungal, and is commonly found on the backs of many amphibians, and particularly protects them against pathogenic infections such as the chytrid fungus. I thought this was particularly interesting as when I was studying down in Peru, I was working with a PhD student who was studying the chytrid fungus down in that area. So very interesting to see that this certain bacteria is present in those areas where the chytrid infection is also prevalent. So it could be a very important species that is present down in that area that's protecting some of those amphibians. So a little summary of my AIM-1 here. I did see a noticeable difference in biodiversity at first glance. There was greater speciation in the deforested areas. I didn't see any pigmentation in the village garden samples, which could be indication maybe of nutrient poor soils or other factors that might change with deforestation. And there was also a larger diversity of antifungal ability in the village garden samples. So I thought that was interesting as well. And then in conclusion, we could potentially be killing some important species with deforestation, such as the Ganthinobacterium levitum that could be protecting these amphibians. So maybe there's not any particular important antibiotic bacteria that we could use, but we're definitely potentially killing some bacteria that are helpful to some of our amphibian species. So my M2, I did look at for actinomycetes, as I had mentioned, for medicinal usage. Actinomycetes are a very dense genre or group of bacteria. They're filamentous and they're fibrous. They have the ability to puncture the agar versus just growing on the surface. So I did use a mixed ester cellulose membrane to isolate for actinomycetes specifically. There's many kinds of bacteria that can grow on the surface of it, but only those that are able to puncture the membrane and grow underneath are the ones that are left to grow after removing the membrane. So this is how I did isolate my actinomycetes. From there, I did select 14 species for studies against other pathogenic bacteria to see if they had antibiotic ability against those. I did add the orange secreting pigment bacteria from my biodiversity assay to that 14 species, so I ended up testing 5 different species. The reason I added the bacteria from the biodiversity assay is because it did show a lot of characteristics that would support that it's an actinomycete. So I did throw that in there with these assays as well. If you look, I did fight against Klebsiella pneumoniae, which is a pathogenic respiratory infection, which didn't give me any antibiotic ability throughout any of the 15 actinomycetes. Streptococcus pyogenes is a fever-causing strep throat, and I did get a couple killing zones produced by actinomycete number 7, which gave us about a 3 millimeter killing zone, and actinomycete number 11, which gave an impressive 10 millimeter killing zone against the S. pyogenes. When putting against methicillin-resistant Staphylococcus aureus, I did also get 2 hits here for MRSA from actinomycete number 9 and actinomycete number 11. Once sequencing that, again, what I did is I only sequenced the actinomycetes that showed some antibiotic ability. So I sent a total of 4, and of those 4, I only received clean readings from 3 of them. My actinomycete number 6 matched 99% similarity to a genus Streptomyces. And Streptomyces is the #1 genus of actinomycetes that produces a number of the antibiotics that we currently have. I do suspect that it's very similar to actinomycetes species A3, and that description based off of the publisher, they did describe it as having a brown diffusible pigment, as you see in actinomycete number 6 there. Number 7, interestingly, gave me an 84% similarity to genus Bacillus, which is not an actinomycete, but did appear very similar to number 6, again having the brown diffusible pigment, so that was interesting to see that there were 2 completely different genuses. And then my actinomycete number 11, which was found in the biodiversity assay, matched about 97% similarity to Streptomyces species CS2 2012, and I'll talk a little bit about that here. The actinomycete number 11, or the orange water-soluble pigmented actinomycete, did show, again, the antibiotic ability against S. pyogenes and the MRSA. Specifically, those are gram-positive bacteria. When weighted against the gram-negative bacteria, Klebsiella pneumoniae, I got nothing. So it could indicate that this particular bacteria is maybe disrupting the building of the gram-positive cell wall in those 2 bacteria. So looking at identification, it is roughly 97% similar to the Streptomyces species CS2 2012. The criterion for identifying and classifying a microbe as a new species requires a 1 to 1.2% difference in the 16S rDNA sequence, which is typically about 1,500 base pairs long. My particular sequence differed at least 2.6% or more as compared to the closest identification. So it could suggest that the species that I found is a unique species that I found in my pristine samples down in the Amazon. So a summary of my aim-2, there's many species of actinomycetes that are present down in the Amazon. I chose to study 15. There were much more than that present based off of my screens for the actinomycetes. there's potential antibiotic species that are there that are unidentified at this point. Not just my actinomycete number 11, but also some of the other actinomycetes I had did not match anything 100%. So it could indicate that there's many species there that are unidentified. And so specifically for my actinomycete number 11, more studies need to be performed, such as which compounds is that secreting that is causing that antibiotic ability, and is it effective against other gram-positive bacteria and not against other gram-negative bacteria, and essentially is it going to be safe for humans? Is it something that we could use for antibiotics in medicine? So some of my acknowledgments, my advisor, Dr. Gary Combs, who's in the back there, the Alpha Chi National Honor Society, I did get a research grant for that, so they did help fund me there, as well as the Better Future for Iowans grant, did receive some funding from them, and just Iowa Academy of Science for hosting me. So, my references. Any questions?
Wait, oh.
Yeah, you got one? Sorry.
Yeah,
I like to answer.
When did you go down to Amazon?
Last June. So I've been working on this project for a little over a year with preparation and doing things. So
Did you go back and forth?
I went one time. Yep, and I obtained soil licenses. My advisor obtained them, and my name was placed as a carrier. So we were able to take samples back from across the borders.
So you brought them back for all the process?
Yep.
You didn't do the process?
Nope, I didn't actually do my research down there. I did field research down there, and I obtained the soil samples there and brought them back to the United States. Worked in our lab at Waldorf. So.
How many village gardens did you go? I mean, I saw a number, I forgot what the number was.
Well, unfortunately, I was only in one specific village. So that's another thing that made it somewhat difficult, was that because we were in a national reserve, there was very few places that had been deforested. So I had one specific village that we had obtained permission from to take some samples from their gardens. So I had about 2 separate gardens from their village. So it's the closest I could get to deforestation. So, but I mean, it still serves the purpose. So, yeah. Any other questions? Yeah.
I got a question for you. That Xanthinobacterium that's on the back of amphibians. Okay, so if we clear cut the forest, maybe we're killing off that microbe in the soil. You suppose the amphibians would be a reservoir for future reinoculation of the soil?
Could be. I guess it would depend. I guess amphibians aren't naturally in deforested areas anyway, because if you're taking away their habitat, you're taking away their moisture, you're taking away the places that they live. So essentially, if you were to get the pristine area back, then yeah, they could possibly transmit that bacteria to that area. But with deforestation, if we completely take away all of those plants and vegetation, it's completely going to get rid of that microbe if the amphibians don't return to that area.
Thank you.
Any questions?
Okay, thank you again. Thanks everyone, that concludes our session. Thanks for coming.